There are several theories about the limits of human lifespan, although they have not been fully elucidated by science. At present, the limit of human lifespan in its natural state is considered to be 115–125 years. However, advances in medicine and biotechnology can lead to people living to be over 150 years old. In the future, if technology that can fundamentally control aging appears, further life extension may not be a dream. Statistical life limits. The oldest officially recorded person in the world at the moment is Jean Calment of France, who lived from 1875 to 1997, achieving a staggering age of 122 years and 164 days. There is still no solid evidence beyond this record. From a statistical point of view, the upper limit of human lifespan is generally considered to be around 115 to 125 years. In particular, a study published in the journal Nature in 2016 proposed the theory that the maximum human life expectancy converges to about 115 years. The study provided a new perspective on longevity and sparked a debate about the limits of longevity. As our understanding of human lifespan grows, Calment's record remains unique, and it will be interesting to see how future research will change this limit. Elucidation of the mechanism of human lifespan may also contribute to the extension of healthy life expectancy, and future scientific exploration is expected. The oldest person in the world is Jean Calment, 122 years old, and this photo was taken when she was 40 years old. The life of Jean Calment. Jean Calment is known as the longest-lived person in history, living to 122 years and 164 days. She was born in Arles, France, in 1875 and grew up in a wealthy family. He married at the age of 21 and had a daughter, Yvonne, who died at the age of 36. As a secret to his longevity, Calment incorporated olive oil into his diet and also used it on his skin. He also liked wine and chocolate, and even when he was over 100 years old, he was active and rode a bicycle. When she was 13 years old, she met the painter Vincent van Gogh and said he was a crusty person. At the age of 90, when selling her home to a lawyer, she entered into a contract to transfer it after her death, but the lawyer died before her, and Calment continued to receive the profits. It is believed that her longevity was influenced by moderate exercise, diet, and optimistic personality. Limits of biological lifespan. The limits of human lifespan are deeply related to cellular aging. A typical factor is telomere shortening. Telomeres are structures at the ends of chromosomes that shorten slightly with each cell division. Telomeres have the role of protecting genetic information from damage, but when they are shortened to a certain length, cell division stops and cell senescence occurs. This limit is called the Hayflick limit, and it is believed that normal human cells reach the end of their lifespan after dividing about 50 to 70 times. This acts as a cell safety device and also prevents unlimited growth. However, Some cells can repeatedly divide by activating an enzyme called telomerase and repairing telomeres. The continued proliferation of cancer cells is related to the abnormal activity of this telomerase. Recent studies have shown that proper regulation of telomerase may help to suppress aging and prolong lifespan. Here is an image of telomeres. The double helix structure of DNA and the telomeres at its ends are depicted as glowing, expressing the association with cellular aging and health. Here is an image of telomerase. It emphasizes the molecular structure of enzymes and their relationship with DNA, and expresses their role in cell lifespan and aging. Possibility of extending life through technological innovation. In recent years, advances in medicine and science have drawn attention to the possibility of significantly extending lifespan. Gene editing technology, CRISPR, is particularly promising. By rewriting specific parts of DNA, this technology can suppress genes that accelerate aging or repair damaged genes. For example, research targeting genes such as P16 and P21 involved in aging is progressing, and it has been shown that it may suppress the decline in cell function due to aging. Next, cell regenerative medicine (iPS) cells is expected to artificially produce pluripotent stem cells from the patient's somatic cells to repair aging organs and tissues. For example, it may be applied to restore the function of the heart and brain that have declined with age. In addition, nanotechnology is being used to remove waste products and damaged molecules in cells that cause aging, and to rejuvenate cellular function. If these technologies are put to practical use, the lifespan of more than 150 years may become a reality. Here is an image depicting gene editing technology. It depicts the precision editing of DNA with CRISPR-Cas9 and the futuristic atmosphere of the lab. This is an image of iPS cell therapy. In a futuristic lab, scientists are using iPS cells to study organ regeneration. Gene editing medicine researcher. Researchers who are particularly famous for their work on CRISPR, a gene-editing technology, are Emmanuel Charpentier and Jennifer Doudna. Charpentier elucidated the basic mechanisms of the CRISPR-Cas9 system and developed a way to apply this technology to gene editing. She has received many awards for this achievement. On the other hand, Doudna developed the CRISPR-Cas9 technology in collaboration with Charpentier, which greatly improved the accuracy and efficiency of gene editing. She has also received many awards for her contributions to the advancement of CRISPR technology. In addition, Emmanuel Charpentier and Jennifer Doudna were jointly awarded the Nobel Prize in Chemistry in 2020. Their work has had a significant impact on the innovation of gene editing technology and its medical applications. CRISPR technology is expected to have applications in a variety of fields, including the treatment of genetic diseases, cancer research, and crop improvement in agriculture. They are Dr. Emmanuel Charpentier and Dr. Jennifer Doudna, who won the Nobel Prize in Chemistry in 2020. iPS cell researcher. A particularly famous researcher in the study of iPS cells, induced pluripotent stem cells in cell regenerative medicine, is Shinya Yamanaka. In 2006, Professor Yamanaka published a method for creating iPS cells from mouse skin cells. The study showed that the cells were reprogrammed to become pluripotent. This achievement has revolutionized regenerative medicine and disease research. In 2012, he was awarded the Nobel Prize in Physiology or Medicine for this discovery. Since iPS cells can differentiate into various cells, they may be used for the treatment of neurological diseases, heart diseases, diabetes, etc. Patient-derived iPSCs can also be used to create models of specific diseases that can be used to develop new treatments. Professor Yamanaka's research continues to have a significant impact on the future of regenerative medicine. Professor Shinya Yamanaka presents his Nobel Prize in Physiology or Medicine. Now, what do you think? What do you think about the limits of human life expectancy?